This is a production of Cornell University.
I'm going to start us off with talking a little bit about myself and where I come from and the people that I come from. So I grew up in a trailer at the edge of the Tonto National Forest in New River, Arizona. So think desert wilderness like down here. This is my first day of school ever. Um, I'm an enrolled member of the Oglala Lakota Nation in South Dakota. That's where my family and I would spend a lot of summers in the house that has been in my family for over 200 years and on homelands for time immemorial before that. My mom is from Bon Po, Thailand. My mom's sitting over there. It's where we get the Po part of our names. And through a long and winding journey that I won't fully get into here, that was filled with a lot of adventure, a little bit of being homeschooled, and a small stint of being homeless and living in a tent. My family and I moved to Hawaii, and that's where Ed mentioned. I got my bachelor's degree at the University of Hawaii at Hilo, and after that, I moved back to one of my homelands to get my master's degree at South Dakota State. I'm the first person in my family, or my mom's family, to have gone to college, and the first person in all of my family to get any sort of graduate degree. And I'm not telling you this because I want you to feel sorry for me or think differently about me or my talk. It's just that given my life and my cultural background, I feel like a statistical anomaly every time I enter academic spaces, and especially here at Cornell. And so I know I was never meant to be here. But I hope that today I can tell you a little bit about what I've been able to do in the past 6 years and how much this means to the people and places that I come from. So for the past few years, I have been working on maize, and maize is a fantastic species to understand the genetic basis of complex traits. And so maize is an annual outcrossing species that's diploid with 10 chromosomes, and globally it's grown on around 200 million hectares. And it serves as a fantastic system to study the genetic basis of complex traits, to learn about gene regulation, and then also validate all of those findings within the field. And so my project is split into 3 different parts. The first is on elucidating the patterns of pleiotropy. The second is on the regulatory impact of TEs, and the last is on field testing. And so for the first on pleiotropy, it's helpful to start off with some definitions because pleiotropy is still really confusing even to me. So pleiotropy is a single locus that can affect multiple different seemingly unrelated traits. And there's a lot of different ways that this pleiotropy can manifest itself. The first is with biological or horizontal pleiotropy. That's where you can have a single causal variant with a star that then impacts 2 different traits. You can have mediated pleiotropy that where one trait is then causal for another. And then you can have spurious pleiotropy, where regions of really strong LD make it hard to distinguish what causal variant is leading to what trait. And in maize, there are a few examples of verified pleiotropy, and these are things like teosinte branch 1, one of the major domestication genes that impacts lateral branching, inflorescence length, and branch length. And there's also one of the major flowering time genes, VGT1, that impacts flowering time, leaf number, and plant height. And so looking across larger sets of genes and not only domestication or flowering genes, the only real pleiotropy that has able to be found is within traits sharing the same developmental unit. And so things like tassel architecture and ear architecture, like inflorescence traits. There's also some pleiotropy between flowering and leaf traits where you need to have a certain number of leaves before flowering initiates and then that life cycle finishes off. And there's further evidence of this with sorghum and maize, where the only pleiotropy that's really found is in very few SNPs, and they are typically between, again, things that are sharing the same developmental unit. Within a lot of the human genetic studies, they've been able to look at things like how pleiotropy is more associated with more highly expressed genes that are expressed across more tissues, along with DNA accessibility and DNA conservation. But in maize, a lot of these things are largely unknown. And so in this project, there are 2 main questions where what are the actual patterns of pleiotropy? Do they kind of mimic some of these large effect domestication or flowering time loci, or are they different in large standing natural variation populations? And the second is, can pleiotropy be modeled? And if so, what kind of biological and noise terms actually explain the amount of pleiotropy present? And so in order to do this, we collected every single published phenotype within the Maize NAM and 282 populations, and this came out to be over 120,000 different traits, and they were for things like RNA expression values across different tissues, metabolite data, and also field traits, so like the traditional plant height, kernel row number, and inflorescence architecture. And so with these traits, we performed GWAS using our tassel. And then for our pleiotropy metric, we counted the number of unique traits mapping to each interval. And so what this looked like for interval A, for example, there's 2 shapes that map to it. And so it's 2 unique traits. For interval B, it would just be 1. C is our least pleiotropic interval, so it's 0. And then E is our most pleiotropic interval with 3 unique traits mapping to it. In order to get a kind of null baseline of what no pleiotropy would look like, we permuted our traits anywhere from 5 to 10 times, ran GWAS again, and then got our null counts of what no pleiotropy looked like. And so for all the plots that I'm going to show you, all the types of pleiotropy are mixed up. We didn't try to distinguish between any one of these, and so it's just a mixed bag of everything. Realize I could turn the captions off. Okay. So to prime you for the plot that I'm going to show you, this shows on the x-axis the log number observed over permuted traits, and the y-axis is the number of intervals. And the main thing is that anything that falls along the dashed line means that there's no pleiotropy. Anything to the left of the dashed line means that there's noise, and anything to the right means there's some. And so for the NAM field traits, around 1% of intervals actually show evidence for pleiotropy, and it controls around 5 different traits. Looking across all the other populations and trait types, this number varies from 1 to 32% of intervals, controlling anywhere from 5 to 400 traits. And even though this seems like a lot, in later slides, I'll show you that this is primarily due to noise terms imparted through the GWAS process. And so now that we know that there is variability in pleiotropy, we bring back a lot of the terms that have been important within the human genetics models, like gene expression, DNA accessibility, DNA conservation, and some noise terms that are imparted through GWAS, like the mean amount of LD within the interval we're looking at, the size of the interval, and then also the number of input GWAS SNPs that we used. And we put all these things together and build a random forest model to determine what are the most important variables in explaining pleiotropy. And this shows that primarily statistical noise terms imparted through GWAS are the most important. And so again, these are things like SNPs per interval, the physical size, and the average amount of LD. Of much lower biological importance are things like sequence conservation, RNA expression, or protein expression. And a very similar trend is shown across all the other populations and trait types that we looked at. And so altogether, LD and noise terms explain pleiotropy much better than any other biological feature or term. And we hypothesize that most descriptions of pleiotropic genes in the past are probably really confounded by linkage, because it's a hard thing to break out of, and there's a lot of it. And so the key takeaways from this project were pleiotropy is not as common as we previously thought, and the 1 to 32% of pleiotropic intervals that we found can originate from sites with varying chromatin accessibility, expression, and conservation levels. Most observed pleiotropy is noise, and so LD will really mess up any pleiotropy estimates that we get. And last, pleiotropy in crop plants is highly constrained by evolution. And so the low to modest levels of pleiotropy actually kind of make sense, because it would allow for selection to more easily fine-tune and adapt varieties to different locations. And I like to think about, if you had a single gene that controlled 50 different traits, it's much harder to work with that single gene as opposed to 50 genes controlling 50 different traits. And so even though pleiotropy might have had little genome-wide impact, the impact of trait curation and the GWAS results themselves had a really large impact. And I've been lucky to be a part of numerous different papers that have used these traits and phenotypes to predict traits with RNA expression values, or look at how they can be explained by conserved non-coding regions upstream of genes. So that was good. The next project that I worked on was looking at the impact of TEs on gene expression. And so a study I was a major contributor to in 2021 showed that around 34% of the expression variation in genes can be explained by cis-regulatory variants. And these are variants that occur within genes or right next to genes. And one of the longstanding questions has been, how much do transposable elements or TEs themselves contribute to this gene expression variation? And it kind of makes sense in maize because 85% of the genome is made up of transposable elements, or these repetitive sequences. And a famous example of this is the bronze 1 locus in maize, where the different rows correspond to different maize inbred lines, and the yellow bars are retrotransposons, and the orange or the red triangles are DNA transposons. And you can see that the maize genome is absolutely littered with transposable elements throughout its intergenic space. In terms of TEs that contribute to gene expression, teosinte branch 1 is another really famous example where a hopscotch retrotransposon brings in an enhancer that then increases gene expression by around 2 times. And this leads to the differences in plant architecture between teosinte and maize. And bringing in an enhancer is just one way in which a TE can potentially impact gene expression. You can imagine with the bronze 1 example, you could get a TE that inserts into an exon and leads to a loss of function of the gene entirely, or it could be even more complicated than that, and the TE could introduce methylation to the surrounding region and silence the gene entirely. And so for a large complex trait like yields, TEs have been shown to impart really subtle effects on fitness. And this was another study I was a part of that's now on bioRxiv that showed that individuals who had more TEs had showed lower grain yields in the maize-NAM hybrids. And making a number of assumptions on per plant yields and planting density and the number of kernels per ear, this came out to losing around 1 to 8 kernels per additional 14 megabases of TEs. And the question kind of still remains, though, if these same small effects occur for smaller molecular phenotypes like gene expression, like they do for yields. So the key questions for this project were, do TEs impact gene expression within the maize inbreds and hybrids? And if so, at what distances do these effects occur at? What TE families cause the largest effects? And if so, are there any known patterns among those families? And last, how much expression variance do these TEs actually explain? And do they mimic some of the large domestication loci, or are they more subtle like yields? And so to do this, we use the high-quality maize-NAM genome sequences that were released a few years ago. And we also collected around 372 RNA samples from both the maize inbred founder parents and then also hybrids between B73 and 2 other testers. We collected RNA samples from across 4 different tissues and quantified our expression counts with salmon. And then for our TE annotations, we used the ones that were generated as part of the NAM genome project. And so they were the EDTA TE annotations. And so to actually model the effect of TEs on expression, we chose to calculate the number of TE base pairs in non-overlapping windows upstream of each gene. And so for this example, even though it looks weird, the gene transcribes over to the left. I know it looks funny, but I'll explain the other parts of the model and it'll hopefully make sense. There we go. Oh no, it really squirrels. Okay, so we know that there's TEs upstream of each gene, and we also know that there are, or DNA methylation plays a large role in whether or not genes are expressed. And we know that unmethylated genes tend to be expressed. And so we layer on unmethylated regions on top. And then we go window by window and count the number of TE base pairs occurring within that window and the number of unmethylated regions also occurring in that window. And we do that for every window upstream up to 15 kb away from genes. And then we do that for all the other genes that are present in the genome. And so we take all of those model components and then we build a model that actually asks what is the effect of expression with TE base pairs and the number of unmethylated regions. And so this plot is showing the gene. There we go. Oh wait, no, it's not showing. Okay, it's showing the gene that again transcribes to the left and multiple distances going away from that gene. And the yellow line shows the count of unmethylated regions and the effect estimate on the y-axis from the model. And we could see near the core promoter of genes, near the core promoter, unmethylated regions are really important. And that makes sense because unmethylated genes tend to be expressed. In terms of TEs, we find a weakly negative effect of TE base pairs when they insert within the core promoter, along with another more weakly negative effect near the actual promoter of genes. And this kind of suggests that their TEs are imparting subtle negative effects when they insert into core regulatory regions. And that makes sense because they might be potentially deleterious. One of the main problems, though, with this model is that it's looking across all TE families, and all of these families differ really greatly by their insertion preferences, their actual sequence content, and the also physical size of the TE itself. And so this is an example just looking at the differences of TE length on the y-axis between DNA transposons and retrotransposons in Maize. And retrotransposons or retros tend to be much larger in general than DNA TEs. Kind of looking at this plot in a different way in my data, this plot is looking at the proportion of TE families that fall into 4 different size categories based on their cumulative abundance across NAM. And so for very large TE families, the ones on the bottom, there's only 48 of them in my data set that make up, that are over 100 megabases in NAM. And they actually make up like 50% of the NAM genome. But there's an overwhelming majority of really small TE families out there that still could potentially cause effects, even though they don't contribute a lot of total base pairs. And so in order to see if there was an effect between smaller TEs and really large TEs, I grouped them into these 4 different size categories and ran the same model as before, seeing if there's an impact of gene expression and TE base pairs given in each one of these small families. And so this is just looking at one DNA transposon in the top 5% of TE families causing the largest effects on gene expression. And this is a DNA TE, a helitron. And so we can see along distance, there's a variable effect of this one TE family along distance. And if we look at the other small families, they tend to cause the largest overall absolute effects on expression versus much larger families, which are in all of the different colors here. And so overall, yes, we do see an effect with TE size, and we also see an effect by distance. And so we're still looking into what a lot of this means and whether or not it's just highlighting like the TE family insertion preference into these different regions, or if it's actually something more biological. And so all of our past models have shown that distance is really important in determining if a TE has an effect or not. And so we wanted to run a more simplified model just with distance to see what that effect was. And so we took the number of base pairs until the first insertion from a particular family. And so this one would be the first mint TE, the first blue TE family, and the first orange one. And then we do this for all the other TE families and first insertions of that family. And so this is what our model looked like. We just took expression against distance for each family separately and did the same model for each family separately. And this plot is looking at the median distance to that family by the partial adjusted R squared on the y-axis of how much variation it actually explains in expression. And around 1 kb away from genes tends to have the largest effects on expression. I should also explain that the color of the points, refers to the number of genes that TE was present near. And so the largest effect TEs tend to be present in very few genes overall. TEs that are present in more than 1,000 genes don't tend to cause any large effects whatsoever. And so the top 5 families that contribute to these effects tend to be things like helitrons and other DNA TEs, along with one LTR retrotransposon. And a lot of the future work in the next coming months is going to be towards validating some of these effects and trying to determine if there's any patterns in the genes that they insert near. So the key takeaways from this project, TEs in known regulatory regions decrease gene expression. And they tend to be near the core promoter or actual promoter of the gene. And these are known regions already that are enriched for open chromatin or TF binding sites. So it makes sense that TEs would impart more of a negative effect there. Next is that small TE families tend to have the largest effects. And last is, TE families only explain a fraction of the variance in the cis regulatory expression in the small subset of genes that they actually insert near. And this suggests that the Maize genome might be using selection to restrict large effect TE insertions in favor of more small effect ones. And so most of my work until this point has shown that quantitative traits are highly polygenic and that any candidate loci that we find will have incredibly small effects. And a lot of this is dictated by Alan Orr's work and along with Fisher's geometric model that says as a population gets closer to its fitness optimum, such as Maize, the smaller and smaller those effects will actually be. And this makes sense because Maize has undergone domestication already. And so any trait that we're looking at now will have really small effects overall. And so for yields, the most complex and quantitative of all traits, we wouldn't expect anything different. And this segues into my last project on yield testing that was published in Nature last year. So this is sparked by a lot of papers that have been published over the last 20 years that say that they can increase yields by anywhere from 8 to 68% through a single gene. But one of the main problems is that these results never translate into real yield gains on farm. And after reading a lot of these papers and running a structured review of them, I found that they have a lot of common problems among them. And these problems are things like a misunderstanding of what yield actually is and how yield components contribute to that. This also has to do a lot with measuring single plant yield versus plot yields. Next is not matching on-farm conditions. So a lot of folks are not testing in elite germplasm and having confounding edge and density effects that lead to misleading yield estimates. There's also questions of scale with hand harvesting versus mechanically harvesting these experiments. There's also insufficient environmental and plot replication. And so there's a good number of greenhouse-only studies. And this typically isn't a problem if you're trying to do like a proof of concept experiment. But when you're saying, 4 plants grown in a greenhouse are going to change the face of agriculture forever, it starts to become a problem. So, yeah. There's also very little plot or environment replication and testing for yield effects in only a single year. There's also a lot of testing genes that might have already been fixed within breeding programs, and also looking at yield effects in alleles that have been segregating within breeding populations for the past few decades. And often when you read really closely within these papers, you find that there are a lot of trade-offs with other yield components in those yields. So it's good to think about when you bring those particular yield alleles to fixation. And the last is lack of controls. And so a lot of folks don't have empty vector controls or don't backcross their varieties into non-transformed varieties or elite varieties to kind of clean up some of those background epigenetic effects that can occur during the transformation process. And so I talk about the latter two within my paper, and you can read about those there. So I am just going to dive a little bit deeper into the first three. And so for a misunderstanding of yield and yield components, yield is typically the weight of the dry grain or the dry matter content of the storage organs harvested per unit area. And so what a lot of these papers tend to do is measure differences, for example, in grain length, for example, between a wild type and a mutant, and say that that increase in like 1/4 of a millimeter of grain length is then going to translate into plot-wide yield later on. The same goes for changes in grain length or differences in grain fill between a wild type and a mutant. Another one of my favorites is measuring biomass changes in Arabidopsis and saying that that will then translate into crop plants. And so it was only, or 40%, or that's a lot of them, of the studies in my review measured yields within single plants or measured yields components and passed that off as their yield increase. One of the main problems with this, though, is that increases in individual yield components or single plant yields won't necessarily scale, especially it doesn't imply that yield per area will increase. And this is beautifully exemplified in a lot of Donald Duvick's work, looking at the year of hybrid introduction and the pioneer maize breeding program from starting in the 1930s up to the 2010s. And it shows that when a variety is planted at incredibly low density, older hybrids tend to yield just as much as newer hybrids. However, when you increase the density, older hybrids are incredibly outcompeted by newer hybrids in general. And this implies that single plant yield hasn't really changed over time. However, the yield per unit area has, and that has been a selection target in maize breeding programs over the past century. And kind of scarily, only 16% of the studies in my review actually planted at commercially relevant densities. Quite typically, they're at incredibly low densities, and that will skew yield estimates in and of itself. Up next is insufficient environment and plot replication. And so this is an example from wheat, looking at the wheat or yield within a plot, starting from the center and going out to the edge. And you can see as you go out further to the edge, those yield estimates will increase because those plants along the edge have more access to light and nutrients and all those good things. And so they'll yield more. Typically what folks do is only measure the stuff on the inside. And something else that can impact just plot yield in general is competition between plants. And so a lot of studies don't tend to actually have border rows or put really competitive genotypes next to each other, and that will skew a lot of these estimates. There's also small plots in terms of the physical size of the plot and the number of individuals that can lead to unreliable data. And a whopping 92% of the studies had experimental designs that would amplify edge effects. So it's something to keep an eye out for. And yeah, very few studies actually measured yields within the center rows. And for example, in a 4-row plot, discarding the outer 2 rows and only harvesting the center. Some last quick stats on this. In terms of plants per plot, it varied from anywhere from 4 plants to 900, and 900 is approaching the scale of a small scale yield trial, but still, there's around 40% of the studies measured less than 20 plants. In terms of plot replication, 46% of them measured in less than 3 plots overall, and typically this was only in a single year. And in terms of multiple year testing, only 63% of them tested in less than 3 years of testing. So very little validation often goes into some of these large high impact papers, which is kind of scary. In terms of not or limited testing in elite germplasm, this is an example from tobacco, looking at tobacco yields in a bunch of commercial tobacco hybrids and Petite Havana. And Petite Havana has been used in a lot of studies because it can be transformed, but it's also been used for a lot of yield estimates and some high impact papers. And the funny thing is that Petite Havana yields 1,600% less than its commercially competitive counterparts. So quite the difference. And 46% of the studies did not test in elite germplasm. And so this is a really common one. One of the favorite analogies that I've heard for testing in elite germplasm is like changing a single part in a Model T versus a Ferrari. And so a Model T is not elite or competitive, and so any improvement that you make to it will seem great, but it's limited in its frame and its design by how fast it can go given that improvement. But one of the main arguments that I've gotten is that they're available to academics to actually test. They're transformable, so that's why folks use them. And this is as opposed to something like a Ferrari that's elite, it's commercially competitive, and so any improvement that you make to it will already add to a optimized engine and design. But for these, typically they're only available to folks within industry or breeders themselves, given patent variety protection. And outside of plant breeding, most folks don't know that X patent variety protected lines exist for them to test and actually validate effects in. And I would kind of refer to these as kind of like Guy Fieri's car in diners, dive-ins, and drives. They are fairly competitive lines, and so any improvements that you make in yields can be realistically tested in somewhat quality germplasm. And the best part is that they are available to academics to test in, so it's a good ticket. So bringing all of these things together, I grouped a lot of the things that I talked about into 3 general categories. And so they're things like controlling for moisture. Did they avoid edge effects in their experimental design? Did they replicate their experiments? And did they test an elite germplasm? And if the study did that thing correctly, I gave it a green box. If they didn't do it well, I gave it a black box. And if it was unclear, if that information wasn't provided, I gave it a different color. And so looking across studies, no one thing consistently goes wrong within them. Each of these studies shows a variable experimental design and quality, and so it's really important to check for a number of different things when evaluating the quality of these experiments. And it's important to note that no single yield gene has ever been commercialized, and it's not from a lack of trying. And this is as far as we can tell in the public data, even though 2 genes have gone through regulatory steps, and I'll talk about one of them in the next slide. But this is one of my favorite papers within this space. It's from Carl Simmons, and it looks at a 2-decade-long industry breeding program where they tested 1,600 genes from 47 different species at scale in the field. And they looked at things for growth and yields, like water use efficiency, nitrogen use efficiency, all different kinds of traits. And they found that of these 1,600 genes, only 8 of them significantly contributed to yields. All the rest were either non-significant in gray or significantly drugged down yields in the environments that they were tested in. The only gene that has shown a lot of evidence and has gone through regulatory approval has been ZMM28, and it's a maize domestication transcription factor that across most of the environments and conditions it was tested in increases yields by around 2%. And one, this is extraordinary because you could statistically actually test for a 2% yield gene, but then the second thing is that this gene actually exists, which feels very rare, very modest, but rare. And so given that a lot of these single yield genes consistently get published in high impact journals, I found it necessary to write more of a policy piece to ensure that yield testing is more robust going forward. And to do this, I assembled a team of global domain experts from across the fields of quantitative genetics, plant breeding, molecular biology, and population genetics to write this perspective piece. And I'll say right now that all of these yield testing criteria aren't novel. They're all written about in Fisher's 1935 design of experiments. They're published in Kieselbeck's PhD dissertation from the University of Nebraska-Lincoln in 1917. So they've been around for a while. And those are back in the day when planters used to be drugged behind horses and they could still make plots well. And so the criteria that we came up with were things like using standard definitions of yield. And so this is measuring plot yield versus single plant yields. This is testing for genotype by environment by management effects. It's matching on-farm conditions. And so this is thinking about plant competition and density and edge effects as you're creating your experimental blocks. And it's also following on-farm practices for planting and harvesting and fertilizer applications. There's also prioritizing genes that plant breeding may have missed, or checking to see that they're not already fixed within commercial breeding programs. Using appropriate controls and elite germplasm, and so testing those empty vector controls and wild types. And then also developing collaborations, because no one person knows how to do all of these things. And to kind of conclude all of these different projects in a mixed bag of a slide, anywhere from 1 to 32% of intervals show some sort of pleiotropy in maize, and most of this is noise imparted through the GWAS process. For TEs, we found that primarily small TEs that insert into known regulatory regions lead to the largest negative effects on gene expression. And for yields, we redeveloped a set of yield testing standards to robustly test for effects in the field. And hopefully this will help researchers and reviewers in the future critically analyze this body of work. And a couple key lessons that I learned through my PhD, I split this off into more scientific and personal ones. But the first is I can never escape the constraints of Fisher and Orr. They are 2 people that are always there for me, and they always bring me small effects. A lab motto has been fail fast, and I would build on that and say building and nurturing the strength of fail fast is really, and trusting yourself in that process is really helpful to, growth or my growth and development as a scientist. And also, I've used social media a lot, especially Twitter, as a platform for positive change within our scientific community to, one, call for more rigor within yield testing studies that led to the Nature paper, but then also serving as, probably some grad students reviewer number 2 and correcting all of their yield things. And the second is calling for more cultural change within our scientific societies that will hopefully end up leading to better environments for our Black, Brown, and Indigenous scientists. In terms of personal things, I found that community is really important, and I'll talk about mine in the next slide. They're the folks that help keep you sane throughout this process. And the last is you yourself are the most important project that you will ever work on during your time at Cornell. And no scientific project is worth your mental or physical well-being. And so we're all here to develop and to learn and to grow. And so as long as you yourself are doing better than you yourself yesterday, you're already winning. In terms of finding community, I've been really lucky to be a part of Synapsis and the Indigenous Grad Student Association over the past few years that have provided a lot of great friendships and sounding boards for things. I'd also like to say thank you to my family for supporting me this entire time and through this very stressful process. So I'm very thankful for them. I'm also thankful to a lot of the mentors that have helped me along the way. And these are the folks that taught me genetics and statistics, folks that have helped me through a lot of different projects and encouraged me to get into some of my first research experiences, as well as folks that have helped me move on to the next stage of my career and find jobs, along with my mom, who's filming all of this from the back, supporting me this whole time. I'd also like to say thanks to the mentors that are no longer with us. My master's thesis advisor, Don Auger, passed away in 2021, and he always said that for this talk, he'd drive here and sit in the front row, and so that's why there is a seat with the blanket reserved for him. And last but not least, I'd like to thank the Buckler Lab for all of their help throughout various projects. My committee, Cedric and Wojtek, along with the American Indian and Indigenous Studies program. If not for them, I wouldn't have written my last 3 papers or dissertation, and so very thankful for them. And I have been funded over the past few years by a NEFA predoctoral fellowship, and that's been great. And so with that, I'd be happy to take any questions.
Question and a comment. I wonder if you're finding that pleiotropy plays a smaller role than folks previously thought. Is related to the fact that maybe we can now dissect phenotypes better, like we understand what a phenotype is and what are distinct phenotypes then.
Yeah, I think a lot of it has to do with the type of association models that people were using before, because a lot of pleiotropy was called the QTL studies, and that just makes it seem like a whole chromosome arm is significant, as opposed to GWAS that gives you a likely list of candidates that are causal. And so, yeah.
And my comment is that actually it is also a kind of form of a question, that the last bit that you talked about. I wonder to what degree you think that these other extending of validity of results of scientific papers is driven by the people who write them or the editors of journals that push folks, particularly these highly visible papers. If I was to place the blame, my suggestion would be 80% editors, 80% people who submit to pressure. I wonder what your thought is, since you actually very carefully read all those papers.
Yeah, I think it's a little bit of a mixed bag. I think a lot of the responsibility goes to editors because they are responsible for finding the correct peer reviewers for these kinds of articles that typically these yield studies tend to only be like a couple of sentences or a table in the supplementary info. And so they typically try to find reviewers that can cover the majority of the paper versus a couple sentences. And I think that's a lot of the reason why these things can get published eventually.
But they're also attractive.
Yeah, they are also highly cited and really, yeah, well used. So it's mixed. Yeah, dear.
Superb talk, thanks. I had a related question to Wojtek's about publication. I mean, plant genetics as a whole has just churned out massive volumes of GWAS studies. Most of those hits don't get used, and it's always felt like a part of that is that a GWAS fits in a dissertation and a variety doesn't. 12 years to make a variety, 3 years, field trial plus analysis and writing for a GWAS. Same with QTL mapping before it, and test out 5 GS models after it. Have you thought, as somebody who's thought like really heavily and looked at these and looked at 25 of them very intently and thought about them intently, I mean, do you have thoughts on, one, if you feel like that statement I just made is true, and then two, if you have thoughts about how in a field where start to finish, the process takes 12 years, nobody wants to stay for that for a PhD, how we could mitigate that and start having more things that are relevant to variety development, but that fit within a 5-6 year time span.
Yeah, it's a little hard.
Solve the field.
I mean, in the second version of our yield paper, like we talk about ways in which to describe your results in a way that's not like this gene will change the future of agriculture and increase yield by like 50%. It's more modest in that like future or additional testing is needed before it can be used in the field, or talking about it differently with industry partners, for example, who have the better abilities at scaling this. And so in that way, you still get a paper that's still important and show the importance of a gene. But yeah, it's hard when the cycle of academia is really short and dependent on a grad student or a postdoc, and it's hard to say where the responsibility lies in some of these larger projects. But I would say like, write about the results a little bit differently and go from there. Oh, yes. And Mark says he's going to use this paper in his class. That's cool. I'm sorry to future grad students.
It already is being used at a number of universities right now.
Yeah. No questions in the chat.
Very good.
Yeah, so the genes that are essentially having no relationship to yield, are they, and this might be more of a speculative question, but are they genes that are not actually scaling up and have no relationship to yield? Or do you think that they are genes that have something that is relevant, but only to that scaled down experiment? For instance, maybe that field did not have enough water, and so it's actually a drought related gene that actually increased.
I think they're typically sometimes only good in some of those smaller experiments. There have been a number of studies from industry looking at how variable these single gene effects are by environment, and they show just gigantic G by E effects. And so for any one of these genes to actually go through the regulatory process, it costs like hundreds of millions of dollars in order to do all the safety, all of the yield trials in different locations. And so whenever an industry company wants to commercialize one of these genes, they need to make sure that it's stable across the majority of environments that it's in. And so that's why ZMM28 is one of those rare exceptions of like, they're not willing to pay $200 million for a one environment, one condition kind of yield gene, as opposed to something that will work well across a lot more. Hopefully that answers your question.
Can I make a comment as opposed to asking a question? I just wanted to say that there's an uncommon amount of wisdom in the talk that you just gave, as opposed to results and cool analyses or whatever. It's a different order of things. And so my comment has to do is one that I want everybody to hear. I'm a little scared about saying this, but there are two things that you said that are really strong. One thing that you said was, I was never meant to be here. And the other thing that you said was, cultural change is a really important thing. So I just want to make sure that our audience is hearing that, and thank you.
Thanks.
Questions?
Oh, yeah.
Yeah. Thank you for the talk. I'm kind of interested about your take, like what could be the evolutionary role of pleiotropy and transposable elements, in your opinion? Like, are they more on the back seat or kind of the driver of that opportunity?
I think they might lead to some of the larger adaptive changes, especially TEs themselves, because some of these effects have led to some of the really large domestication genes, for example, or radical changes in gene expression, especially under like stressful conditions like salt or drought. A lot of TEs can get activated. And so I think there is a really large impact, even though right now for things like yields, it doesn't seem like it. And as for pleiotropy, I still think it exists. I still think it's out there, but a common limitation to what we're doing is that GWAS can only look for pleiotropy in common variants because you need like segregating alleles. You could still have pleiotropy at fixed loci like TD1 because you have a lot more safety in fixation. You can't lose critical functions if that gene gets lost or the allele frequency changes and it's no longer prevalent in the population. And so it still has some effect, but not massive. Oh, we don't need bonus slides. Okay. Here's your thoughts on candidate gene identification could be refined for relevance to elite germplasm and breeding programs. Often we rely on insights from GWAS and very diverse association panels, which typically don't have as much applied relevance in structured breeding programs. For that, I'm not sure. Sometimes going to other species and finding genes that haven't already been tested for a long time within the same species can help. Some of even the yield genes that have been published before do have a lot of promise because sometimes they're introduced from different species and you can have novel effects that haven't been tested. So maybe there as a start. Hopefully that answers your question.
Let's thank us for Merritt providing us wisdom.
This has been a production of Cornell University, on the web at cornell.edu.